Scientists across the world who are working on synthetic genome expressed some of the difficulties while working in lab. It's difficult to understand about the fundamental language of the genome. Every genome rewrite program so far has grappled with substantial and unexpected challenges, and the era of made to older genome remains out of reach. In evolution, genome modification is very fast due to high mutations as well as biological and abiotic stresses. It includes biotic as well as abiotic problems. Coming to the basics, most synthetic genome projects are top down efforts that take a naturally occurring organism and pare away or redesign its DNA. That provides a valuable initial framework relative to bottom up approach in which the goal is to build a working genome from scratch. When it comes to tinkering with genomes, the margin for error is surprisingly slim. If you create an error in the essential gene, you are going to wipe the organism out. So, taking one gene at a time is very important. First time when researchers published their 2016 report on their first minimal cell, nearly one third of the cell's remaining genes had no known functions. Means almost one third, 149 to 473 genes were unknown and their functions were not known. But gradually, now in 2023-24, it reduced up to 80 to 90%, means only 10% is remaining unknown. To determine which genes were necessary, both the projects used random mutagenesis, basically introducing untargeted perturbations throughout the genome, and asked which ones the cells could tolerate and which one severely determined cellular viability. Many bacterial genes have multiple jobs, making it difficult to recognize which is the essential function. Example, enolase, that's an enzyme with a well-known role in carbohydrate metabolism that also turns out helps to degrade unwanted RNA. Minimized genomes. To avoid the difficulty in the initial stages, scientists opted minimized genomes. Mechanistic models with machine learning to predict the consequences of genome manipulation across a broad range of biological functions. These are described by thousands of interlinked equations, yielding blueprints for bacteria that have 40% fewer genes than their wild type. For example, in E. coli, 40% reduced genome they prepared. Protein coding sequences are built of nucleotide triplets known as codons, with 61 possible codons for 20 natural occurring amino acids as well as 3 stop codons that terminate protein synthesis. There is considerable redundancy in the resulting code. Various teams shown that by comprehensively converting each instance of a given codon to one of its synonyms, one can repurpose the codon. Example, in E. coli strain called Ocre, in which 2 stop codons were reassigned to direct the incorporation of the non-natural amino acids para-acetyl 1 phenylalanine and one more. These amino acids have chemical properties and functions that don't exist in nature, but recoding can also serve as firewall that prevents the interaction and exchange of genetic material with other organisms in natural environments. Let's see, genome recoding requires much planning and effort. After researchers have found all instances of the codon they wish to eliminate, they must then work out how to replace it without disrupting the affected genes or regulatory machinery. Bacterial genes often contain regulatory sequences in the protein coding sequence. A gene on one DNA strand can overlap with a gene on opposite strand. Seemingly minor changes can thus have major unexpected consequences. Some changes disable existing regulatory elements or unwittingly create new ones. Others established new protein coding sequences. The minor challenges, till now we saw what are the major challenges, but the minor challenges are like technical challenges, cost of large scale precision DNA synthesis remains high. Example, commercially synthesized DNA building blocks of up to 10 kilobases might cost roughly $0.10 per base to produce. But many eukaryotic genomes contain repetitive elements that are hard to synthesize, can easily double the cost of the starting material. And the time consuming. Researchers who spent 10 to 12 years, even many spent 5 to 6 years, but the duration taken is too high. Then how to deal with this problem? In the current era, we are dealing with the artificial intelligence and machine learning. Algorithmic tools can also help the researchers to model and predict the outcomes of some genome rewriting experiments in advance. That is called ground truthing. Means preparation or ground truthing is always important to start any of the project. One more thing is we should take the small and self-contained like bacterial genomes are ideal test beds for developing synthetic genomic tools. Example, E. coli, which has a single circular chromosome of roughly 5 million base pairs, the Cerevisiae genome encompasses more than 12 million base pairs across 16 linear chromosomes. That means we should start with a small genome. The take-home message: at a crossroads, the synthetic genomic field is at a crossroads. Whereas many researchers plan to dig deeper into their model organisms of choice, others are eyeing new terrain. Sophisticated design algorithms with greater predictive power could cut these costs by genetically more accurate genomic blueprints that streamline the testing and optimization process. Example, several groups have demonstrated means they used artificial intelligence for constructing functional DNA molecules that are based on pattern learned from vast training sets of sequence data. It's not science anymore, it's literally a black box. It hopes to see progress in building mechanistic machine learning models that are trained on well-defined, carefully annotated experiments, but this is a slow and costly process. It estimates that models for complex eukaryotic genome are probably about 25 years behind. That means instead of depending on the old sequenced genome, we should take the newer one which are functionally annotated. In the early days, you would just try to write an app and compile and hope that there is no error. But once you get past that first stage of hello world, the next phase will be much more intention driven design. That means instead of finding a needle in haystack, you should go for particular thing. That means your goal should be a material specific or goal specific, then only you can achieve something. That is the take home. Thank you. If you have any doubts, keep it in the comment section. Keep on subscribing, commenting, sharing. Thank you once again.